Hello and welcome to our first VCE Biology online remote learning classroom. Today we'll be diving straight on into Area of Study 2 as a part of Unit 3 and beginning to look at antigens as well as pathogens. As usual, we have our learning intentions that we should be able to achieve as our presentation completes. So today, by the end of our little recording today, we should be able to define terms such as disease, pathogen, and explain briefly what the immune system is able to do and what it means. We should be able to look at what antigens are and then maybe even be able to list some examples, and then hopefully begin to link this to some idea of what we call pathogens, defining ideally some of their cellular features and being able to tell differences between them by what these differences are. Finally, we'll aim to be able to then define what a leukocyte is as a cellular example of a cell involved in the immune system, and then be able to explain how this is then related to B and T lymphocytes. So let's start off with some of the basics, I guess, that underpin this knowledge that you might have learned or remembered from other years, but just some of the basics that we'll get into here when we're talking about the diseases. Diseases are usually defined as being conditions that hinder the normal function of an organism. So diseases can be infectious, and ideally this means they're caused by a particular agent, or they can be non-infectious, so diseases that might be caused by environmental or genetic factors. So for example, when you're thinking about your infectious diseases, you're thinking about diseases that you can catch from someone else, and your non-infectious diseases, you're thinking about something that you might get potentially from the environment, or more commonly, something that might be genetic. So not something you're technically getting from something else, but maybe something you inherit in your genes. Infectious diseases are caused by particular organisms or agents called pathogens, and pathogens we define as being agents that cause diseases. They can be cellular or non-cellular. One of the reasons here that we've specified that they're agents that cause disease is because they're not really all organisms. Some of them aren't technically alive, because usually one of the defining characteristics of something that's not alive is that they are made of cells. And we do have some pathogens, some agents that cause disease that are non-cellular, are not composed of cells. Continuing on with some background knowledge, we've just got here that the immune system is our bodily system that responds to disease and pathogens. It is composed of a variety of innate molecules, specific cells, and entire organs. So I want you to think that your immune system is this really complicated set of different parts around your body, from really small molecules that flow around, in particular fluids such as your blood, to actual cells that defend you against disease, to entire organs whose whole function it is to try and keep you safe and healthy. To be able to respond to disease, the immune system needs to be able to recognize pathogens, and that's going to move us on to our next point where we begin to look at antigens. Speaking of antigens, these are our unique molecules that can be recognized by the immune system. Antigens can stimulate immunity and can also be referred to as being immunogens. They are commonly protein structures that are found on the surface of the plasma membrane, but they can also be lipids, carbohydrates, nucleic acids, those biomacromolecule families we've spoken about before. Some examples of antigens might include the glycolipids or glycoproteins we've spoken about being shown on the plasma membrane, toxins secreted by pathogens. These are just harmful sort of chemicals that are released by these agents that cause disease here, these pathogens, and proteins in the flagella just being the tail of a bacterium here. So really what we want to be thinking when we're thinking about antigens, these are our unique molecules that identify our pathogens, and they allow us to be able to see what the pathogen looks like. They allow our immune system to be able to detect and recognize when a particular threat or something that's going to cause disease is within itself. Of course, antigens aren't just unique to being, to belonging to a particular pathogen. We also have what are called self-antigens, and these are of our host organism. So on our cells, we have particular molecular markers that tell our immune system that this is us, and then there's no need to respond. But also when we have a pathogen inside us, these represent these non-self-antigens, and those are of a foreign material or of a particular pathogen. In most cases, the immune system is able to recognize self-antigens and does not respond. We don't want our immune system causing inflammation or causing some sort of damaging response to our own body. That would be quite harmful. But obviously, and you guys might have heard of some sort of autoimmune diseases, and these are when the immune system does respond to particular self-antigens, and then we begin to have some sort of problems here. So just going over quickly this diagram, so we have here these little markers here, these would be our self-antigens, and we can see our little immune cells were reacting positively when we see our own self-markers or our self-antigens, and we know that this is self. Contrastingly, we have our immune cell here, and it's not happy, tell by its comical face, to these non-self antigens that are present on this particular looking bacterial pathogen. Sometimes in our immune system we can develop some sort of errors, and this can be particularly when our body begins to recognize self-antigens as non-self antigens, and this may lead to damage of particular cells or tissues, and this is usually, as mentioned, described as being an autoimmune disease, where our immune system is attacking our own self cells and based on those own self-antigens. One of the examples that you might have learnt about or might remember from Unit 1 biology is in type 1 diabetes, in which the immune system responds to self-antigens on the beta cells in the pancreas. So these cells normally produce insulin, but due to the immune response, the antigens are then recognized, and then the immune response to those cells then leads to them being destroyed, leading to an overall lower production of insulin in the body, and then a real misregulation of the amount of blood glucose levels in that particular host organism. Now that we've spoken about the antigens, those molecular markers that allow us to identify particular pathogens and cells within our body, let's begin to actually look at some of those pathogens. So our first group of pathogens we'll be looking at are bacteria, and bacteria are our unicellular prokaryotes that exist almost everywhere. You know, we've got bacteria living on our skin and in our guts and on most of the food and surfaces we eat, and they all come in a variety of shapes and sizes. You can actually see that down the diagram. We've got these circular ones here, we've got these rod-shaped ones here, and we've even got some other chains and different shapes and sizes shown down here. Most of these bacteria are actually called commensals, meaning they live in us and they are even mutualistic in their relationship with us, meaning they can actually be really good for us. But obviously some of them do actually contribute to disease. We actually do require some bacteria to help us synthesize some important molecules, but some bacteria are opportunistic, meaning that they actually can cause diseases in particular environments or on a particular locations. So when we're thinking about pathogenic bacteria, we're usually thinking about them causing disease in an opportunistic manner. So this means when they either get into a different, maybe a different part of the body that they're not normally found in, they begin to then cause disease. Some of the other important things that we might talk about when we think about bacteria are that they classically have this shape here. And this is the shape that maybe you'll see in a lot of exam sort of diagrams and scenarios. You've got this sort of oval elongated shape here with a little flagella tail. Some of the other features that you might see in this particular diagram are that they have plasmids, which are their genetic material. They have cytoplasms like we do. They have ribosomes like we do, but a little bit differently, of course. They have a plasma membrane and usually what's called a capsule and a cell wall. A capsule being a layer of fats surrounding their already carbohydrate-rich sort of cell wall that goes around here. And this is usually the shape you'll see in particular cartoons and diagrams of what bacteria look like. Again, bacteria, these are our unicellular prokaryotes, and they're everywhere, and they usually cause disease in an opportunistic manner. Just one other thing that I'll add talking about bacteria here are some classic examples of bacteria that you might have heard in day-to-day life, such as E. coli or maybe even Staphylococcus, even a strep infection if you've ever had streptococcal pneumonia. Our next group of pathogens we'll be looking at are our fungi, and these are a broad group of unicellular and multicellular eukaryotes, with some examples being yeast and mushrooms that you might have already heard of. They possess a thick cell wall similar to plants, but lack chloroplasts, so they lack the ability to photosynthesize, that being one of the big differences between them in a cellular manner and animal and plant cells. They often cause disease by secreting some sort of digestive enzymes, and these then break down the particular matter that they're then causing the disease. Usually our body recognizes polysaccharides or glycoproteins on the surface of their plasma membrane as an example of their particular antigens that then go on and stimulate our immune system. Some of the diseases you might have heard about that are caused by fungi are things like athlete's foot, for example, as well as some particular STDs. Our next group of pathogens are called our viruses, and these are our singular particles that are composed of some sort of genetic material, either being DNA or RNA, that is enclosed within a group of proteins, and these proteins usually form a coat called a capsid. The capsid actually representing usually a type of recognized antigen that our immune system then recognizes as being a virus. Singular virus particles are usually called virions, and you can see here I've got some examples of visual sort of demonstrations of what some different viruses look like: the HIV, the hepatitis B, Ebola, adenovirus, influenza, and bacteriophage. Another virus, quite famously, I guess you guys will be learning all about and hearing all about right now, is the coronavirus. But something that all of these viruses share is that they can't actually reproduce without a host organism. So remember, these viruses, they're singular particles. They're not actually made of cells, and they're much, much, much smaller than normal cells. And so these are actually also called obligate parasites because, again, they can't reproduce without a host and are not considered alive because they can't reproduce, and they're also not composed of cells. So these little viruses, usually how they cause disease, they go inside a particular cell, they take over their transcription and their translation, and they basically as a little parasite, use that cell to be able to replicate and then form more and more viral particles of themselves. And after replicating within a cell, they usually then sort of replicate to the point where the actual cell then begins to burst or lies down here, causing damage to the particular organism. Our next group of pathogens are actually a subclass of our viruses, and these are called our viroids. They are composed of a singular circular strand of a nucleic acid, and unlike our viruses, they're not actually covered by any sort of protein coat. So really they're just these free floating strands of DNA, usually in this particular sort of circle here. They're not alive, they're obviously not cellular because they're just RNA or DNA, and they are usually and most commonly cause disease in plants, usually by disrupting some sort of protein synthesis. You can sort of see down here. And now for our final major group of pathogens here, we've got our prions, and these are our smallest group of pathogens. So where we started with our bacteria and our fungi, where they were large cellular pathogens, then we moved on to viruses, which are essentially just proteins wrapped around some genetic material, and then our viroids that were just genetic material, and now we're looking at prions, and these actually don't even have any genetic material at all. They're simple just proteins that are different forms of proteins that already exist within the nervous tissue of particularly larger organisms. So really our prions, these are our particular types of proteins, and they're usually based off an organism, and these are just proteins that are just folded in a sort of a weird, really strange different way to particular other proteins. They cause similar proteins to fold into the same shape as the prion, and this misfolding can then cause the tissues or the parts of the body that are affected by the particular disease to lose their structure and their function, and this is the cause of particular diseases such as Khrushchev-Yakov disease. And just one more thing about prions here, you see I've managed to actually get this diagram to work. You can see we've got our prions here causing other similar prions to misfold, and this is just going to change again. So this is our original prion causing other similar prions to misfold, and this is really what we're talking about when we're talking about the pathogenesis of these particular prions. So these misfolded proteins cause other similar folded proteins to turn into themselves, which then allows them to then go on and misfold other proteins, overall reducing the functionality of the particular function of the proteins and the surfaces and structures that they're affecting. So these might be proteins in the plasma membrane of cells, maybe in the brain, such as the case of Khrushchev-Yakov disease, where then these proteins, once they've been misfolded, really don't have the same sort of structural integrity as the particular proteins in the plasma membrane when they're in the normal shape, and then this leads to those particular cells becoming a lot less stable, and this is actually what causes Khrushchev-Yakov disease, or maybe you might have heard of the bovine example of mad cow disease. Now finally, just some of the other pathogens that you might come across in some sort of more out there exam questions, or maybe even in your textbook. We've got these protozoans. These are unicellular eukaryote parasites that usually require a host to reproduce in, being the definition of a parasite. Some of the examples that you might have heard of are Plasmodium, and this is a little parasite organism that causes the disease malaria that I'm sure many of you have heard of before. The next one, and excuse my pronunciation, I do believe this is Umicites. These are protista microbes. These are eukaryotes that commonly cause diseases in plants. We've also got worms such as your tapeworms, your flukes, your roundworm as well, and these are large multicellular eukaryotic microbes that act as parasites you can get from other particular sources. And finally, these are our last group of pathogens. These are arthropods, so these are large invertebrates. Again, these are obviously eukaryotic if they're invertebrates, such as ticks or lice or even mites. Now moving away from pathogens, we'll go back to looking at some of the aspects of the immune system. So within our immune system, we have this bodily fluid, blood and lymph, and in these particular fluids, we have particular cells that whose role it is to combat these diseases and these pathogens that we've been speaking about. The broad term for these cells in these fluids that fight these diseases are called leukocytes, and their roles in the immune system vary and can involve anything up to recognition and response. They might be involved in activating and communicating between other leukocytes, and maybe even establishing immunity, which we'll cover more in depth in a couple of weeks. One of the subgroups of our leukocytes are our lymphocytes here, and these cells are involved in establishing immunity, which as a definition is the long-term resistance of particular diseases and towards a particular condition. So one of the examples that you might be a bit more familiar with is one of the reasons we don't get diseases such as chickenpox or something like that over and over again throughout our life is that we've developed immunity to that particular disease. Some examples of lymphocytes that we have in our bodies are The two different types of lymphocytes that we'll be looking at are our B and our T lymphocytes. So to start with our B lymphocytes, these recognize the antigens that are on the surface of our pathogenic organisms. So you can see that here, and typically when we're talking about our B cells, we're talking about these lymphocytes being involved in recognizing the antigens that are on the actual pathogens that we've just spoken about, those bacteria, the fungi, the prions, and so on and so forth. Our T lymphocytes, in contrast, now these actually look at the antigens that are on the surface of our own self cells. So these are actually recognizing our cells, the host cells, and these are actually usually involved in detecting when maybe these cells become infected by some of the pathogens that we've talked about. So again, our B lymphocytes, again lymphocytes being a subgroup of the leukocytes that we just talked about, involved in some sort of immunity, and our B cells are the ones that recognize pathogen antigens, and our T cells are the ones that recognize the antigens that are found on our own self cells. Obviously, these cells need some sort of way in which to be able to recognize these particular antigens. So both the B and T lymphocytes have what are called receptors on their surface that enable them to be able to detect these particular antigens. The B cell receptors bind and recognize the non-self antigens on the pathogens that we've spoken about, whilst the T cell receptors actually bind to what are called major histocompatibility complex proteins, which are found on the host cells. So these are sort of molecular structures that have our own antigens on them, and these are what are recognized by the T cells, just reinforcing that idea that the B cells recognize the non-self antigens on foreign material such as pathogens, whereas our T cell receptors in on our T cells, these recognize the antigens that are loaded up into these major histocompatibility complex proteins that are found on our own host cells. Upon binding to the antigens, some response is then induced, and we'll talk about this later in a few more weeks. Finally, we'll talk about what happens when our B lymphocytes actually come into contact with a piece of foreign material or a pathogen. So these B lymphocytes produce proteins that are called antibodies. You might have called antibodies before. You might also bump into this little word here, these immunoglobulins or Igs. This is just another term for these antibodies. So these antibodies are proteins that we've talked of, and they're very similar to the receptors that B lymphocytes contain. So these are produced by our B lymphocytes, and they're free floating in the bodily fluids such as our blood and our lymph. They can bind to antigens just like other receptors, and the binding of these antibodies to the antigens on the pathogen usually immobilizes them. So if you can imagine these little sort of dots here, these might be some sort of pathogen that's trying to cause some disease, and you can see they're all stuck together and they're all attached to each other by these different antibodies that we've got here. And so these antibodies have all stuck to them and sort of bound them all together, and in this way they actually can't move around, and it really impedes their ability to cause disease. So this is how B lymphocytes, as an example, are able to actually respond to pathogens, and then by releasing these antibodies to bind to them. And that concludes our first remote learning online classroom for VCE Bio this year. I really hope you've enjoyed this little presentation. Just to reinforce, please at the end of this, make sure you're able to go over our learning intentions and make sure that you're able to do the things that are on screen at the moment. So things like making sure that you're able to define disease and pathogen, as well as what antigens, both self and non-self, are, and some of the cellular features of the major pathogens that we talked about today, such as bacteria, fungi, viruses, prions, the viroids, and such. We also then spoke about leukocytes and lymphocytes, so please do make sure you're able to maybe even list some examples and some of the broad things as to what they do and what are the main differences between our B and T lymphocytes. I look forward to presenting in the future, where we'll be talking about different parts of the immune system, including how our body responds in an innate manner. Thanks for listening.